most powerful electric field your body maintains. 25 million volts per meter, stronger than a thunderstorm, exists inside the membrane of your fat cells. When temperature drops, that field is deliberately collapsed. The entire electrochemical gradient, built at significant metabolic cost, is discharged directly as heat through a single protein channel, producing nothing but warmth. Body built a resistor into its own power plant and calls it a furnace. There is a reason the body's first response to cold is not heat production, but blood redistribution, and the physics of that redistribution follows a law where halving a vessel's radius reduces blood flow to 1/16. There is a protein in brown fat mitochondria that bypasses the ATP turbine entirely, converting the full energy of the proton gradient into thermal output with zero chemical capture. And there is data showing that the thermogenic organ that declines with age can be reactivated, rebuilt by the stimulus it was designed to respond to. All of it comes later. Start with the problem. Start with the problem, because maintaining a constant body temperature in a variable environment is one of the most difficult thermodynamic achievements in all of biology. Mammals hold core temperature at 37 degrees Celsius in environments that range from -40 to +50. Second law of thermodynamics governs the difficulty. Heat flows from hot to cold, and the rate of flow is proportional to the temperature differential between the body and its surroundings. In a cold environment, the body loses heat faster. To maintain a constant core temperature, it must generate heat at precisely the rate it loses it, a continuous balance of thermal input and thermal output adjusted in real time across every condition the environment presents. Body achieves this through two simultaneous strategies: minimizing heat loss through vasoconstriction, insulation, and postural changes, and generating additional heat through three distinct thermogenic systems: shivering, brown adipose tissue activation, and the baseline metabolic output of all cellular chemistry. Each system has different physics, a different speed of activation, and a different capacity. Together they constitute one of the most sophisticated thermal management systems in nature. Control center for this system sits in the preoptic area of the hypothalamus, a cluster of thermosensitive neurons whose firing rate changes precisely with temperature. Warm-sensitive neurons increase their firing rate as temperature rises. Cold-sensitive neurons increase their firing rate as it falls. Balance between these two populations generates a continuous error signal. The difference between actual core temperature and the biological set point of 37 degrees. When core temperature drops half a degree below set point, the error signal triggers three simultaneous downstream responses: sympathetic vasoconstriction of peripheral blood vessels, activation of shivering programs in skeletal muscle, and noradrenaline release onto brown adipose tissue. Precision is remarkable. Core temperature is maintained within approximately 0.2 of a degree Celsius, despite enormous variation in ambient temperature, activity level, and metabolic state. Think about that, the same principle that governs a household thermostat. Compare actual temperature to set point. Generate a correction signal proportional to the error is running in a cluster of neurons in your hypothalamus with a precision that no manufactured thermostat approaches, continuously, without calibration, for an entire lifetime. The engineering is identical. The execution is incomparably better. Now, the first defense. Because the fastest cold response is not heat generation. It is redistribution of the heat the body already has. Peripheral vasoconstriction occurs within seconds of a temperature drop. Smooth muscle in the walls of arterioles supplying the skin and extremities contracts, reducing vessel diameter and restricting blood flow to the periphery. The physics is Hagen-Poiseuille flow. Blood flow through a vessel is proportional to the fourth power of the radius. Fourth power. Not linear, not squared. Fourth power. A 16% reduction in arterial radius, invisible to the naked eye, undetectable by the person experiencing it, reduces blood flow by 50%. Halving the radius reduces flow to 1/16 of the original value. The body's thermal management system operates through radius changes so small they cannot be seen, producing flow changes so large they can be measured on a Doppler ultrasound within seconds. Warm blood stays at the core where the vital organs require it. The extremities are deliberately sacrificed thermally, kept cool to reduce heat loss from the skin surface. This is why hands and feet are cold first. It is not a failure of circulation. It is the physics of Hagen-Poiseuille, protecting the heart and brain at the expense of the periphery. A counter-current heat exchange system in the limbs amplifies the effect. Arteries and veins run in close parallel in the arms and legs. Warm arterial blood flowing outward transfers heat to cool venous blood, returning from the extremities before the venous blood reaches the core. The arterial blood arriving at the fingers is cooler than the arterial blood that left the heart. The venous blood arriving back at the core is warmer than the venous blood that left the fingers. Heat is recovered in transit. The extremity functions as a radiator with a recirculation loop, losing some heat to the environment, but recovering a significant fraction through the countercurrent exchange before it reaches the organs. A person who comes inside from the cold and holds a warm cup of tea is doing more than seeking comfort. They are applying external heat to the peripheral vascular bed that vasoconstriction sacrificed, warming the tissue that the body deliberately cooled, allowing the smooth muscle in the arterioles to relax, restoring peripheral blood flow as the thermal threat diminishes. The warmth that returns to the fingers as they wrap around the cup is vasodilation. The Hagen-Poiseuille equation running in the opposite direction, vessel radius increasing, blood flow rising by the fourth power of the change. The relief is vascular, not merely sensory. And when redistribution is not sufficient, the body activates its largest organ as a dedicated heat generator. Because muscle is thermodynamically inefficient, and in cold, that inefficiency becomes the point. Skeletal muscle converts approximately 25% of ATP energy into mechanical work. The other 75% is released as heat. Under normal conditions, this inefficiency is a metabolic cost. The reason you overheat during exercise, the reason muscles feel warm after exertion. In cold, the body exploits the inefficiency deliberately. Shivering is rapid, involuntary, asynchronous contraction of skeletal muscle fibers designed to produce minimal net mechanical work. The contractions cancel each other out, while maximizing ATP consumption and therefore heat production. Every mole of ATP hydrolyzed releases approximately 30 kilojoules of free energy. In shivering muscle, approximately 22.5 kilojoules of that, 75%, is released as heat. The metabolic rate during maximal shivering can reach 5 times the resting basal metabolic rate, equivalent to moderate aerobic exercise, sustained entirely by involuntary muscle oscillation. The body is running its largest metabolic organ as a furnace, exploiting the thermodynamic waste that physical engineering spends careers trying to eliminate. Every shiver is an inefficient engine running on purpose. The waste heat that engineers try to minimize in machines is exactly what the shivering body is trying to maximize. Same physics, inverted engineering goal. Shivering draws primarily on muscle glycogen, the stored glucose that fuels rapid muscular contraction. At maximal shivering intensity, glycogen depletion in the active muscles can occur within 3 to 4 hours, at which point the shivering intensity declines even if the cold persists. The body has exhausted the local fuel supply of its emergency heating system. Blood glucose can partially substitute, but the delivery rate through the bloodstream is slower than the consumption rate during intense shivering, and the muscles begin to fatigue. This is the thermodynamic limit of shivering. It is powerful, rapid, immediately available, but it is running on a finite fuel tank that empties under sustained demand. Person who has been shivering for hours and then stops shivering, not because the cold has resolved, but because the body has reached its glycogen limit, is in a more dangerous thermal state than the person who is still shivering. The cessation of shivering and continued cold is not adaptation. It is fuel exhaustion. Core temperature begins dropping at that point. But shivering has limits beyond glycogen. It is metabolically expensive, physically exhausting, and it interferes with voluntary movement. A body that relied entirely on shivering for thermogenesis would be a body that could not function in cold. shaking too violently to use its hands, burning through reserves at an unsustainable rate. The body needs a heat source that runs quietly, continuously, without interfering with everything else the body needs to do. Now, the furnace. Because brown adipose tissue is the most extraordinary thermodynamic machine in the human body, and the physics of how it generates heat is almost never explained. To understand what brown fat does, the normal mitochondrial mechanism must be clear first. In every cell, the electron transport chain pumps protons, hydrogen ions, from the mitochondrial matrix across the inner mitochondrial membrane into the intermembrane space. This creates a proton gradient, a difference in proton concentration and electrical charge across the membrane. The gradient has two components, a chemical component from the pH difference and an electrical component from the charge separation. Together they constitute the proton motive force. Proton motive force across the inner mitochondrial membrane is approximately 180 millivolts across a membrane 7 nanometers thick. That produces an electric field of approximately 25 million volts per meter. The electric field inside a thundercloud capable of producing lightning is approximately 100,000 volts per meter. The electric field your mitochondria maintain across their inner membrane is 250 times stronger than a thunderstorm. Every mitochondrion in your body is holding this field right now. Every one of the thousands in every cell, across every tissue, continuously, as a baseline operating condition that the cell maintains at significant metabolic cost for every second of its existence. Under normal conditions, protons flow back across this gradient through ATP synthase, a molecular turbine that rotates physically as protons pass through it, using the rotational energy to drive the synthesis of ATP from ADP and inorganic phosphate. ATP synthase is one of the smallest known rotary motors, approximately 10 nanometers in diameter, spinning at up to 600 revolutions per second, producing approximately 3 ATP molecules per full rotation. The stored electrochemical energy of the proton gradient is captured as chemical energy in ATP through a genuine mechanical process. A turbine spinning inside a membrane at a scale invisible to light microscopy. The system is coupled. Proton flow produces ATP through mechanical rotation. UCP1 uncouples it. UCP1, uncoupling protein 1, also called thermogenin, is a channel protein in the inner mitochondrial membrane of brown fat cells that allows protons to flow back across the gradient without passing through ATP synthase. The proton motive force drives protons through UCP1, and the energy is released directly as heat through molecular collisions. As the protons dissipate down the electrochemical gradient, no ATP is produced. The entire gradient, the 25 million volt per meter electric field that the electron transport chain spent metabolic energy building, is collapsed deliberately, and every joule of that energy becomes thermal output. Result is extraordinary. Brown fat mitochondria can run their electron transport chains at full capacity, burning glucose and fatty acids, pumping protons, maintaining the gradient, while producing zero ATP and maximum heat. The furnace runs at full output. The turbine is bypassed. The controlled collapse of an electric field stronger than lightning generates the warmth. Thermodynamic cost is deliberate and specific. Building the proton gradient requires the electron transport chain to oxidize NADH and FADH2, electron carriers loaded by the citric acid cycle, pumping protons against the electrochemical gradient at each of the 4 complexes. This is expensive work. The cell burns fuel to build the gradient, and then UCP1 collapses the gradient without capturing the energy. In any other context, this would be metabolic failure, a short circuit in the power supply. In brown fat, it is the design. The short circuit is the function. Every calorie of fuel that brown fat burns produces heat instead of ATP, and the heat is the product the body requires. Under a microscope, brown fat cells contain mitochondrial density so high that the iron in the electron transport chain proteins gives the tissue its characteristic color. Brown under a microscope, distinctly darker than the white adipose tissue that stores energy rather than burning it. A biopsy of brown fat looks different from white fat because it is doing different physics. White fat is a battery. Brown fat is a furnace. They sit in the same body, sometimes in adjacent tissue, doing opposite things with identical fuel. You see, every brown fat cell is a power plant, running at maximum capacity with its generator disconnected. The fuel burns, the protons pump, the gradient builds, and then UCP1 opens and the gradient discharges as heat instead of being captured as chemical energy. The cell is deliberately wasting energy, and the waste is warmth. That mechanism, a protein channel that bypasses the ATP turbine and converts the full proton motive force into thermal output, is the finding that made thermogenesis land differently from anything else in physiology for me. Because it means the body did not stumble into heat production as a byproduct. It engineered a dedicated uncoupling channel, expressed it specifically in thermogenic tissue, positioned that tissue around the major blood vessels supplying the brain and heart, and activated it through the sympathetic pathway that governs every other cold response. The furnace was designed. And here is where the furnace sits, because the anatomy matters as much as the biochemistry. Anatomically, brown adipose tissue concentrates in specific locations: the supraclavicular region above the collarbones, the paravertebral region alongside the spine, the mediastinum between the lungs, and the perirenal region surrounding the kidneys. These locations place brown fat in direct proximity to the major blood vessels supplying the body's core: the aorta, the subclavian arteries, the renal arteries. Heat generated by brown fat enters the bloodstream directly and warms the blood supply to the brain, heart, and kidneys before it reaches those organs. Placement is not incidental. Brown fat is a core heating system, anatomically positioned to warm the blood flowing to the organs that cannot tolerate temperature variation. The brain loses function when its temperature drops even modestly. Cognitive processing slows, reaction time lengthens, consciousness narrows. The heart's electrical conduction system is temperature sensitive. The QT interval on an electrocardiogram lengthens as cardiac temperature falls, increasing the risk of dangerous arrhythmias. The kidneys require stable temperature for glomerular filtration rate maintenance. Renal blood flow and filtration decline as temperature drops. Brown fat sits next to the arteries that supply all three, generating heat that enters the bloodstream upstream of the organs that need it most. Newborns illustrate what brown fat was originally for. A human infant cannot shiver effectively. The motor pathways that coordinate shivering are not fully myelinated at birth. The infant's primary thermogenic defense is brown fat, which constitutes approximately 5% of body weight at birth, a far higher proportion than in any adult. The brown fat is concentrated in the interscapular region, around the kidneys, and along the great vessels, maximizing heat transfer to the core blood supply. The infant warms itself almost entirely through UCP1-mediated uncoupling. Shivering develops later. Brown fat is the original furnace. Shivering is the backup that develops when the motor system matures. Activation runs through sympathetic noradrenaline release onto beta-3 adrenergic receptors on brown adipocyte membranes. Noradrenaline binding activates adenyl cyclase, increasing intracellular cyclic AMP, activating protein kinase A, which phosphorylates and activates lipase, releasing fatty acids from intracellular triglyceride stores. The fatty acids do two things simultaneously. They enter the mitochondria as fuel for the electron transport chain, and they directly activate UCP1 by displacing inhibitory purine nucleotides from the protein through allosteric binding. Fatty acids are both the fuel and the switch. The molecules that feed the furnace also turn it on. When fuel arrives, the furnace activates. When fuel is depleted, the furnace shuts down. The control is automatic, molecular, and self-regulating. And then, what happens in the first seconds of extreme cold? Because the cold shock response is a separate system entirely, operating on a different timescale and serving a different purpose. Cold water immersion, or any sudden exposure to extreme cold, triggers a response that begins within 1 second of contact. Cold thermoreceptors in the skin. TRPM8 ion channels, the molecular sensors that also respond to menthol, fire at maximum rate. The signal reaches the brainstem and triggers an immediate sympathetic discharge. Noradrenaline release spikes to approximately 300% above baseline, higher than the noradrenaline response to most acute psychological stressors, higher than moderate exercise, one of the largest acute catecholamine surges measurable in a living human outside of pheochromocytoma. Gasp reflex, an involuntary deep inhalation, is driven by this sympathetic discharge. Breathing rate increases to 40 breaths per minute or more, heart rate and blood pressure spike. The cold shock response is not thermoregulation. It does not produce heat. It is a threat response. The nervous system treating extreme cold as an emergency indistinguishable from a physical attack. TRPM8 channel that initiates it is the channel activated by menthol. The sensation of coolness from mint is an identical molecular event as the beginning of a cold shock response at a vastly lower intensity. The fresh feeling of menthol on the tongue is a whisper of the signal that, at full volume, produces the gasp reflex in cold water. Think about what the gasp reflex means for cold water safety. The involuntary inhalation that cold shock produces is the mechanism by which cold water drowning occurs in the first seconds. Before hypothermia, before exhaustion, before any thermal management system has had time to activate. The person who falls into cold water and inhales water in the first gasp is not panicking. They are executing a reflex that the brainstem cannot suppress. Cold water drowning is not a thermoregulation failure. It is a threat response consequence, operating faster than any thermal defense. Cold shock response habituates with repeated exposure. Subjects who undergo repeated cold water immersions show progressive reduction in the gasp reflex, the heart rate spike, and the hyperventilation. The brainstem learns that the cold stimulus is not a lethal threat and reduces the magnitude of the response. The habituation is specific to the cold shock component. It does not reduce the thermal management responses that follow. The body learns to stop treating cold as an emergency while continuing to treat it as a thermal challenge. The distinction between threat response and thermoregulation, between the first seconds and the minutes that follow, is a distinction the nervous system can learn to make through experience. And here is what happens to the furnace with age, and why the data that follows changes the story. Over the lifespan, brown adipose tissue activity declines over the lifespan. PET, CT imaging using fluorodeoxyglucose consistently shows lower brown fat volume and metabolic activity in older adults compared to younger ones. The mechanism involves multiple converging factors. Reduced sympathetic tone with age means less noradrenaline. Reaching brown fat, declining beta-3 adrenergic receptor density reduces brown fat sensitivity to the noradrenaline that does arrive, and the progressive replacement of brown adipose tissue with white adipose tissue, a process analogous to thymic involution, reduces total thermogenic capacity. By 65, brown fat volume and activity is typically a fraction of what it was at 25. This is one mechanism behind the increased cold sensitivity of older adults. The dedicated thermogenic organ has been partially decommissioned. Furnace is smaller, fuel line is narrower, the thermostat signal is weaker. Well, the data that makes this more than a decline narrative, because the furnace responds to the stimulus it was built for. Cold acclimation studies, exposing subjects to mild cold for several hours per day over weeks, consistently show measurable increases in brown fat volume and metabolic activity in adults of all ages. A research group at Maastricht University demonstrated that 10 days of mild cold exposure, not extreme cold, not ice baths, but sustained mild cold at temperatures between 15 and 16 degrees Celsius, temperatures most people would describe as cool rather than cold, increased brown fat activity significantly in young adult subjects. The subjects did not exercise during the cold exposure. They sat in the cold. The stimulus was thermal, not physical. The brown fat responded to the temperature signal alone. A Japanese group led by Yonoshiro showed that previously brown fat negative subjects, adults with no detectable active brown fat on PET/CT imaging, developed detectable, metabolically active brown fat after 6 weeks of daily cold exposure at 19 degrees for 2 hours. Brown fat appeared where none had been visible. The organ was recruited from precursor cells that were present in the tissue but dormant, waiting for the cold signal that modern indoor environments no longer provide. Tissue can be recruited. The furnace that age and chronic warmth decommissioned responds to cold by rebuilding. The mechanism runs through PGC1 alpha, the transcriptional coactivator that drives mitochondrial biogenesis in response to AMPK activation, stimulating the growth of new mitochondria in existing brown adipocytes, and potentially the differentiation of beige adipocytes in white adipose tissue depots. Beige adipocytes are intermediate cells, white fat cells that, under sustained cold stimulus, express UCP1 and acquire thermogenic capacity they did not previously have. Cold does not merely activate existing brown fat. It can create new thermogenic capacity in tissue that was previously storing energy rather than burning it. Beige adipocyte finding changed what adipose tissue means in the context of aging. White fat was understood as inert storage, a depot for excess calories, metabolically quiet, functionally passive. The discovery that white adipocytes can be converted to thermogenically active beige adipocytes through cold exposure, that the boundary between storage tissue and thermogenic tissue is not fixed but responsive to environmental signals, means the body retains the capacity to expand its furnace throughout life. The raw material for new thermogenic tissue is the white fat that replaced the brown fat during involution. The replacement tissue contains the precursor cells for the very function it displaced. Think about what this means for the 65-year-old who feels cold more easily than they did at 40. The increased cold sensitivity is real. It is the convergence of 5 separate physical changes operating simultaneously. Subcutaneous fat redistributes with age, reducing the insulating layer in the extremities. Peripheral vasoconstriction efficiency declines as vascular smooth muscle loses responsiveness to sympathetic signals. Shivering threshold shifts. The core temperature drop required to trigger shivering becomes larger, meaning the body tolerates more thermal debt before activating the response. Brown fat volume and activity decline, and the baseline metabolic rate decreases as muscle mass is lost, because muscle is the body's primary heat-generating tissue at rest. 5 changes. Each modest individually. Together they shift the thermal balance significantly. The body is generating less heat, insulating less effectively, and responding less rapidly to cold. The feeling of being cold more easily is the body reporting all 5 simultaneously. Subcutaneous fat redistribution is why the arms feel cold. First, the insulating layer that kept heat in the limbs has thinned. The vasoconstriction decline is why the cold takes longer to leave the hands after coming inside. The arterioles are slower to reopen. The shivering threshold shift is why the body tolerates a deeper chill before the shaking starts. The thermostat's trigger point has moved. The brown fat decline is why the quiet, continuous warmth that once ran in the background is less present. The muscle mass loss is why the baseline warmth of simply being alive, the metabolic heat of resting cellular chemistry, produces fewer kilojoules per hour than it did 2 decades ago. None of them are failures. They are calibration shifts in a thermal management system that has been running without stopping since birth, maintaining core temperature within a fraction of a degree through decades of winters and summers, through fever and recovery, through every ordinary day in between. And several of them, brown fat volume, shivering capacity, peripheral vasoconstriction, respond to sustained cold exposure. The body that encounters cold regularly maintains its thermal defenses more effectively than the body that avoids cold entirely. The system was built to respond to the stimulus. In a climate-controlled environment that never challenges the thermal management system, the system downregulates. It is not breaking. It is responding to the signal it receives, which, in a heated home, is a signal that thermogenesis is no longer required at previous capacity. The feeling of cold, the specific cold that arrives more easily and leaves more slowly than it did 20 years ago, is the body reporting 5 separate physical changes simultaneously. Less brown fat, generating UCP1-mediated heat. Less shivering sensitivity from a shifted thermostatic threshold. less subcutaneous insulation from fat redistribution, less peripheral vasoconstriction efficiency from declining smooth muscle responsiveness, less resting metabolic heat from reduced muscle mass. The feeling is accurate. It is not anxiety. It is not hypochondria. It is 5 thermal sensors and 5 thermal outputs reporting a change balance between heat production and heat loss, and reporting it through the only channel the body has for communicating thermal information to consciousness, the sensation of cold. When a 65-year-old says they feel the cold more than they used to, they are reporting physics. 5 separate mechanisms, each measurable, each with an identified molecular or vascular basis, each contributing to a thermal balance that has shifted since middle age. Furnace built into the mitochondria of brown fat. The one that deliberately collapses a 25 million volt per meter electric field to generate heat is still there. It is smaller. It is less active. And the physics says it responds to cold. The body that feels cold more easily now is the body that built this system in the first place. It has not forgotten how.